Gas chromatography is an analytical method used to analyze the complex compounds in a gas sample or sample that can be converted into gases. Resolution of compounds is due to dissimilarity affinities toward to the stationary and mobile phase held within the column. Each of the compound passes through the column at a different speed, depending on computation between volatile stability and interaction with the stationary phase, and this leads to each compound having a specific retention time. To analyze a mixture of alcohol such as 2-propanol and N-butanol, gas chromatography was performed in this experiment. Retention time of standard sample were then determined and compared with an unknown sample to identify its component. Moreover, the influence of temperature on retention time and peak resolution was investigated in order to gain insight into how temperature affect compound separation in GC.
This is the objective of the experiment, which is first is to determine the retention times of N-butanol and 2-propanol. Next is to identify the compound that is present in a standard mixture based on the TR. And then to identify the components present in an unknown sample, and also to determine the effect of the temperature on this TR and RS.
For these experiments, we use shrink and shear filter together. The shrink is used to inject a small volume of sample into the gas chromatograph. The shrink filter removes impurity to prevent blockage and ensure accurate results. The picker is used to hold and mix the sample or solvent during preparation. The dropper is used to add small amount of liquid carefully during sample preparation. Also, the vial is used to store the prepared sample before injection into the gas chromatography. In this experiment, we use two types of chemical. First, N-butanol is used as one of the alcohol sample for GC analysis. It helps in observing the separation and retention time of a higher alcohol. And we use also the 2-propanol is used as reference alcohol. It allows us to compare retention time and peak behavior in gas chromatography. The standard mixture contains equal amount of 2-propanol and N-butanol. It is used to identify peak and compare retention time in the chromatography. Also, the unknown solution contains 2-propanol and N-butanol in a 2:1 ratio. It is analyzed using gas chromatography to determine the composition by comparing it with the standard mixture.
For the methodology of the experiment, and first of all is for the sample handling. First, the shrink was strained with the sample to avoid any contamination happen, and then the shrink was then filled with slightly more sample than the required volume, and then air bubbles were checked and then were removed by gently tapping the shrink. Next, the shrink was held vertically and with the needle pointing upward, and then plunger was slowly adjusted until the required volume was obtained at our eye level, and then the excess sample was wiped off using a tissue, and then finally the prepared sample was injected into the GC injector. In the GC system, the carrier gas will flow from the gas cylinder through the pressure regulator and the flow controller, carrying the injected sample into the column inside the oven. Within the column, the sample components were then separated based on their properties. So, the separated component then entered the flame ionization detector, which is FID, and then which generated signals that were sent to the computer and then displayed as the chromatogram for the analysis. For the methodology of the experiment, and first of all is for the sample handling. First, the shrink was strained with the sample to avoid any contamination happen, and then the shrink was then filled with slightly more sample than the required volume, and then air bubbles were checked and then were removed by gently tapping the shrink. Next, the shrink was held vertically and with the needle pointing upward, and then plunger was slowly adjusted until the required volume was obtained at our eye level, and then the excess sample was wiped off using a tissue, and then finally the prepared sample was injected into the GC injector. In the GC system, the carrier gas will flow from the gas cylinder through the pressure regulator and the flow controller, carrying the injected sample into the column inside the oven. Within the column, the sample components were then separated based on their properties. So, the separated component then entered the flame ionization detector, which is FID, and then which generated signals that were sent to the computer and then displayed as the chromatogram for the analysis. For the methodology of the experiment, and first of all is for the sample handling. First, the shrink was strained with the sample to avoid any contamination happen, and then the shrink was then filled with slightly more sample than the required volume, and then air bubbles were checked and then were removed by gently tapping the shrink. Next, the shrink was held vertically and with the needle pointing upward, and then plunger was slowly adjusted until the required volume was obtained at our eye level, and then the excess sample was wiped off using a tissue, and then finally the prepared sample was injected into the GC injector. In the GC system, the carrier gas will flow from the gas cylinder through the pressure regulator and the flow controller, carrying the injected sample into the column inside the oven. Within the column, the sample components were then separated based on their properties. So, the separated component then entered the flame ionization detector, which is FID, and then which generated signals that were sent to the computer and then displayed as the chromatogram for the analysis. For the methodology of the experiment, and first of all is for the sample handling. First, the shrink was strained with the sample to avoid any contamination happen, and then the shrink was then filled with slightly more sample than the required volume, and then air bubbles were.
Okay, now we will look at the each part of in gas chromatography machine. Okay, here we have a carrier gas with the gas flow regulator And through it, we have the injector port to inject the sample, and in the oven, we can do have the column, and uh, out of the oven, we have the waste and the detector to pass the result or data into the computer.
Hello everyone, in this part, I will briefly explain how gas chromatography works and the role of each main component. Firstly, an inert carrier gas is supplied to move the sample through the system. The flow controller regulates this gas so it flows at a constant rate, which is important for getting accurate and reproducible results. Next, the sample is introduced to the injector port. This part is heated to quickly vaporize the sample so it can mix well with the carrier gas. A septum is used at the injector to allow the sample to be injected without any gas leakage. The vaporized sample then enters the column, which is the main part where separation happens. Different components in the sample interact differently with the stationary phase inside the column, causing them to separate. The column is placed inside an oven, which controls the temperature to improve separation efficiency and analysis time. As the separated components leave the column, they are detected by the detector. The detector converts them into electrical signals, which are displayed by the data system as a chromatogram. Finally, the carrier gas is safely released through the exhaust.
In the chromatogram that we are currently seeing, two main peaks can be observed. The first smaller peak that is 2.86 minutes represents 2-propanol, while the second taller peak 3.4 at 3 minutes corresponds to N-butanol. The reason 2-propanol appears on the left, which has shorter retention time, is because it is more volatile and has lower boiling point compared to N-butanol. This means it moves through the column faster and elutes first. On the other hand, N-butanol appears on the right, which have longer retention time because it is less volatile and has a stronger interactions with the stationary phase due to its longer carbon chain. As a result, it takes more time to pass through the column, giving a longer retention time. The heights of the peaks represent the amount or concentrations of each compound. The taller peak for N-butanol indicates that there is a larger quantity of N-butanol in the sample compared to 2-propanol. The shorter peak for 2-propanol shows that it is present in a smaller amount.
Next, let's take a look at result 2-propanol at 17 degrees Celsius. In this analysis, we examine pure 2-propanol using a gas chromatography with flame ionization detector GCFID. The chromatogram shows a single dominant peak at approximately 2.989 minutes. The retention time is a result of 2-propanol relatively high volatility, meaning it interacts less with the stationary phase of the column and therefore elute earlier. The height area percentage around 99.9% confirm the sample purity, indicating that only negligible impurities are present. Overall, this analysis demonstrates that the GCFID method is effective at accurately identifying and quantifying 2-propanol.
Next is the results for n-butanol at 70 degrees. So as you can see, the number of peak is only 1, which is the same as 2-propanol. And then the retention time obtained is 3.52 minutes, which is longer than the 2-propanol. And why? And it is because it has a higher boiling point and lower volatility. And also because the stronger interaction with the stationary phase.
Analysis was performed using gas chromatography with flame ionization detector (GCFID). The injection volume we use 1 microliter. The chromatogram shows 7 detected peaks, indicating multiple components in the sample. A major peak at retention time, around 3.50 minutes, dominates the chromatogram. This peak contributes approximately 86.5% of the total peak area, indicating the main component of the sample. Other peaks have a very small area percentage, around below 1%, representing minor impurity. So the conclusion, the sample is mainly composed of one dominant compound with small amount of impurity, demonstrating effective separation by GCFID.
Two peaks having retention time of 2.669 minutes and 2.864 minutes were obtained with the standard mixture at the temperature of 100 degrees. These peaks are for 2-propanol and N-butanol respectively. In comparison to the 70-degree, retention time was shorter for both compounds, which infers that highest temperature is causing the volatile to elute faster from GC column. This is because highest temperature means more volatile of the compound that have less interaction with the stationary phase, resulting in the better separation resolution.
Let's move in for the last result, which is standard mixture at 114 degrees Celsius. In this analysis, a mixture of N-butanol and 2-propanol was analyzed using gas chromatography with a flame ionization detector GCFID. The chromatogram shows two main peaks at retention time of 2.377 minutes and 2.476 minutes, representing N-butanol and 2-propanol. This different retention time occur because each compound has different volatility and interaction with the GC column. The error percentage show that 2-propanol is the major component, while the small peak represent trace impurities. This result shows that GCFID successfully separated and identified the compound in the mixture.
So now we are going into the discussion parts. In this experiment, we can clearly see that the focus is the retention time. So what is retention time? Retention time is characteristic for each compound under specific analytical conditions. However, it is not unique to a compound, and two or more substances can share the same retention time. In addition, retention time is influenced by several operating parameters. For example, in gas chromatography, carrier gas type and flow, temperature, column dimensions, and stationary phase polarity and thickness can affect retention time. In this experiment, different temperature ranges were used. So, there are three sets, which is A, B, and C, were kept at 70 degrees Celsius, 100 degrees Celsius, and 140 degrees Celsius respectively. From the graph that shows in the result, it is proven that the compounds take less time to elute as the temperature rises. As a result, the retention time for N-butanol was 3.520 minutes at 70 degrees Celsius, which is in set A, while the retention time for 2-propanol was 2.989 retention time. That was the result for set A, which is were kept at 70 degrees Celsius. Right now, we are going to talk about the next injection, which is in set B, that were kept at 100 degrees Celsius. The retention time for 2-propanol and N-butanol were 2.669 minutes and 2.864 minutes respectively. Next, the retention time for 2-propanol and N-butanol, which is in the highest temperature, that is 140 degrees Celsius in set C, were 2.377 minutes and 2.76 minutes. So, after we are analyzing the results, we can see that the temperature is keep getting shorter, which is when the temperature is keep getting higher. So based on the result, the standard mixture has an average retention time of 2.755 minutes for 2-propanol and 3.332 minutes for N-butanol.
The retention time of 2-propanol and N-butanol were successfully determined using gas chromatography. The unknown sample was found to contain both 2-propanol and N-butanol. Increasing the temperature decreased the retention time of the compound.
Okay, the first question. State the type of compound which are suitable for analysis using GC. Okay, gas chromatography is suitable for analyzing compounds that are volatile and thermally stable, meaning they can easily vaporize without decomposing at high temperature. The compound should also have a low to moderate molecular weight and be a non-ionic and non-polar or only slightly polar. Example of compounds suitable for GC analysis include hydrocarbon, alcohol, ester, ethers, aldehydes, ketones, and other volatile organic compounds.
For the second question, why is FID a suitable detector for this analysis? The answer is the flame ionization detector FID is suitable for this analysis because it offers high sensitivity for organic compounds, provides a wide linear response range, and delivers stable and reproducible result. It is ideal for detecting carbon-containing compound while being insensitive to inorganic substance, making it reliable for quantitative analysis in gas chromatography.
Alright, for two factors which can increase the efficiency of GC column, first is using a longer column. This is because a longer column provides more surface area for separation, resulting in better resolution between compound. Second is using smaller stationary phase particle. This is because they allow for faster mass transfer and sharper peaks, improving separation efficiency.